Hi guys, thank you for joining me today. I'm going to do a lesson on transport plant and photosynthesis. So if you have been struggling with the year 12 content and how it links to the year 13 content, then why not check out this video? Hope you like it and you find it helpful. Do ask questions if you're not sure at any point. Photosynthetic tissue contains cells specialized to carry out photosynthesis. Palisade mesophyll cells are highly adapted to maximize the rate of photosynthesis. They have many chloroplasts to absorb large amounts of light. They are elongated, so they tightly pack together to maximize the amount of light they're capturing. They have thin cell walls to allow rapid diffusion of carbon dioxide, and they have a large central vacuole to maintain the turgor pressure and keep the cells closely packed. Chloroplasts are the site of photosynthesis in plant cells. The actin-based cytoskeleton helps reposition the chloroplast to maximize light absorption. Photosynthesis uses water and carbon dioxide to produce glucose and oxygen. The glucose acts as a respiratory substrate and the oxygen is required for aerobic respiration. Meanwhile, respiration releases carbon dioxide and water, which can be used again in photosynthesis. This establishes an interdependent relationship between the two processes. The structure of the chloroplast is important for photosynthesis. Evidence for its evolutionary origin includes its double membrane, circular DNA, and 70S ribosomes. These features support the endosymbiotic theory, which proposes that chloroplasts evolved when a photosynthetic prokaryote was engulfed by a larger cell and formed a mutually beneficial symbiotic relationship. Thylakoids are flattened sacs consisting of thylakoid membranes surrounding the thylakoid space. They are stacked to form grana, which are connected by membranous channels called lamellae. These membranes provide a large surface area for the absorption of light because many photosystems, electron carriers, and ATP synthase are embedded in the thylakoid membrane. Their flattened shape creates a small internal space, the thylakoid lumen, enabling the accumulation of hydrogen ions and the formation of a steep proton gradient. So to understand how chloroplasts absorb light, we need to take a step back and we need to remind ourselves about visible light. So visible light from the sun is composed of a range of wavelengths, 400 to 700 nanometers. Now the wavelength is the distance from one point of a wave to the same position on the next wave. For example, peak to peak, or it could be trough to trough. When we see a rainbow, we observe all the colours in visible light: red, orange, yellow, green, blue, indigo, and violet. This occurs because white light entering water droplets in the air is refracted, internally reflected, and dispersed, causing different wavelengths to spread out at different angles and form the spectrum of colours that we see. So photosynthetic pigments then absorb these different wavelengths of light and are embedded in the thylakoid membrane within protein complexes to form what's known as a light harvesting system, the antennae complex. The primary pigments, chlorophyll A, absorb mainly red and blue wavelengths of light and reflect green light, which is why most leaves appear green. Accessory pigments, including carotenoids, xanthophyll, and chlorophyll B, absorb additional wavelengths of light and pass the energy to chlorophyll A. This broadens the range of light that can be absorbed, maximizing the efficiency of photosynthesis. In autumn, chlorophyll production decreases and breaks down more rapidly. As its green colour fades, the colours of the accessory pigments become visible, making leaves appear green, then yellow, then orange, or even red. Photosynthetic pigments can be extracted from leaves and then separated using thin layer chromatography. A TLC plate has a thin coating of silica gel, an adhesive substance, which acts as the stationary phase. A pencil line is drawn approximately 1 centimeter from the bottom of the plate, and then the pigment extract is then applied to the line, 1 drop at a time, allowing each to dry before adding the next one. A darker initial spot indicates a higher concentration of pigment. The TLC plate is placed upright in a beaker containing an organic solvent, the mobile phase. As the solvent moves up the plate by capillary action, the pigments dissolve and travel with it. Each pigment has a different solubility in the solvent and different attractions to the stationary phase. This causes them to move at different speeds, resulting in separation into distinct bands on the plate. Now, we can then work out the retention value for each pigment by using the distance moved by the pigment divided by the distance moved by the solvent, and we call that the solvent front. So if you are given in your exam a diagram that shows a picture like this, you would take your ruler, you would measure from the origin to the solvent front, and you would measure from the origin to whichever pigment they want you to observe. That will therefore allow you to calculate the RF value. Now, it may be that you calculate the RF value for a green pigment to be 0.64 and then conclude that it was probably chlorophyll A, and you can tell that because it shows a bluey green colour, which is what you would expect. However, if you're given a table of results that say that chlorophyll A is 0.63 and you calculated a value that wasn't that was named on the table, then that would be evidence against the conclusion. So be aware that expecting you to know how to calculate your RF value, but also how to then use that to draw a conclusion. Photosynthesis can be divided into two stages. The light-dependent stage, light energy is absorbed by photosynthetic pigments and converted into chemical energy, forming ATP and reduced NADP. Light-independent stage, also known as the Calvin cycle, the products of the light-dependent reaction, ATP and reduced NADP, are used to fix carbon dioxide and synthesize organic molecules such as glucose. So to understand how this happens, we first need to look inside the thylakoid membrane. Inside the thylakoid membrane, photosystems are embedded consisting of these antennae complexes and a reaction center. Two types of photosystems, each with its own reaction center. So there is photosystem 2, PS2, and photosystem 1, PS1. The antennae complex captures light energy and transfers it to the reaction center. When a pigment absorbs a photon of light, its energy is transferred to one of the pigment's electrons. This causes the electron to gain energy and move to a higher energy level, the excited state. The energy can then be passed from one pigment molecule to another until it reaches the reaction center. Once at the reaction center, the chlorophyll A molecules can donate their excited electrons to the electron transport chain, initiating the process leading to ATP and reduced NADP formation. So a photon of light gets absorbed, excited electrons move to a higher energy level, energy is released as those electrons return down to their original energy state. So what's actually happening is quite complex, but let's see if we can figure this out. The photon of light can be absorbed by both photosystems simultaneously, allowing each reaction center to donate excited electrons to the electron transport chain. If NADP is available as the final electron acceptor, then non-cyclic photophosphorylation can occur. Electrons from photosystem 2 travel through the electron transport chain, passing through a proton pump to photosystem 1. Electrons from photosystem 1 are transferred to NADP reductase, which reduces NADP to reduced NADP. This movement of the electrons through the proton pump releases energy, which is used to actively transport hydrogen ions across the thylakoid membrane, creating a proton gradient. These hydrogen ions then diffuse back through ATP synthase, also embedded in the thylakoid membrane, driving the production of ATP. Now, to replace the electrons that were lost from photosystem 2 that went to photosystem 1, photolysis of water occurs. Water molecules are split into electrons, hydrogen ions, and oxygen, which is released as a waste product. If NADP is not available to accept electrons, cyclic photophosphorylation occurs in this process. Electrons from photosystem 1 are recycled, moving through the electron transport chain and across the proton pump back to photosystem 1. The electron flow maintains the proton gradient across the thylakoid membrane, allowing ATP production to continue even without the formation of reduced NADP. So it's important to understand the electron transport chain is required to pump hydrogens against the concentration gradient so that they then accumulate within the thylakoid space. This then results in a electrochemical gradient, and those hydrogen ions will flow down the electrical chemical gradient through the ATP synthase. That is chemiosmosis. So once the light-dependent reaction has occurred, we can either end up with ATP molecules, or if it's non-cyclic photophosphorylation, it's ATP and reduced NADP. The light-independent reaction, also known as the Calvin cycle, takes place in the stroma of the chloroplast. It does not directly require light, however, it depends on the products of the light-dependent reaction, ATP and reduced NADP. A molecule of carbon dioxide is fixed to a 5-carbon compound, ribulose bisphosphate, RUBP, forming an unstable 6-carbon intermediate molecule. This reaction is catalyzed by the enzyme RUBP carboxylase, commonly known as Rubisco. Due to the instability of the 6-carbon intermediate, it immediately splits into 2 3-carbon molecules of glycerate 3-phosphate, also known as GP. Now, each GP molecule is converted into a triose phosphate. This conversion requires 1 molecule of ATP and 1 molecule of reduced NADP. Now, during the process, reduced NADP is oxidized, therefore regenerating it so it can return to the light-dependent reaction to accept further electrons and hydrogen ions. The whole reduction stage requires ATP and reduced NADP, which were produced in the light-dependent reaction. So products from the light-dependent reaction are required for the reduction process in the light-independent reaction. Now, the RUBP used during carbon dioxide fixation must be regenerated as well to allow the light-independent reaction to continue. Now, to achieve this, 5 molecules of triose phosphate are converted back into RUBP. This conversion requires energy which is provided by the remaining ATP molecules. 1 triose phosphate molecule leaves the cycle and is used to synthesize other carbon-containing compounds required by the plant. Now, how do we know this? Well, this was demonstrated by Calvin and his co-workers, who illuminated a suspension of green algae in the presence of radioactive carbon dioxide. Samples were taken at different time intervals after the addition of radioactive carbon dioxide. Chromatography was then used to separate and identify the carbon compounds formed. After 1 second, only glycerate 3-phosphate, GP, contained the radioactive carbon. After 5 seconds, additional molecules were found containing radioactive carbon, including TP, sugar phosphates, and RUBP. And after 30 seconds, the radioactive carbon was detected in many other molecules, such as sucrose, amino acids, and lipids. Now, the rate of photosynthesis can be measured in several different ways. We can measure the volume of oxygen produced during photolysis in the light-dependent reaction. We can measure the volume of carbon dioxide taken up, indicating the carbon fixation in the light-independent reaction. Or we can measure it indirectly by measuring the increase in biomass resulting from the conversion of triose phosphates into stored organic compounds. Now, environmental factors such as light intensity, temperature, and carbon dioxide can affect the rate of photosynthesis. If any one of these factors is in short supply, it will then limit the rate of photosynthesis, and this is known as a limiting factor. Now, we can carry out investigations to look at these limiting factors on the rate of photosynthesis. Now, one way in which we can do that is by limiting the light intensity. Light is needed as an energy source in the light-dependent stage. If light intensity is low, less ATP and reduced NADP are produced, therefore less GP will be converted to TP in the light-independent reaction. At very high light intensities, photoinhibition may occur, and this occurs when the electron transport chain becomes overloaded, leading to the formation of reactive organic species, ROSs, which damage chlorophyll molecules, can damage photosystem 2, and can damage the thylakoid membrane. So light intensity is a limiting factor. If it's too low, what we can see is that the light intensity is limiting the rate of photosynthesis, and then it increases until a point at which it plateaus. When it plateaus, then something else is limiting the rate of photosynthesis. So as you can see from my graph, we have different things that can limit the rate of photosynthesis: carbon dioxide concentration, temperature, or some other factor like the chlorophyll molecules, the photosystems, and the thylakoid membrane, which need to be repaired. Now, if we look at temperature, we know that photosynthesis is a set of enzyme-controlled reactions. If we have low temperatures, we have less kinetic energy, less successful collisions, less enzyme-substrate complexes, therefore a lower rate of reaction. Now, if we increase temperature too high, that can result in more kinetic energy, more successful collisions, a higher rate of reaction, until enzymes such as rubisco can become denatured. Now, carbon dioxide concentration is a really interesting one. When carbon dioxide levels are very low and or the temperature is above 25 degrees C, oxygen can bind to rubisco. Oxygen, therefore, is a competitive inhibitor for rubisco. Now, this leads to the production of a toxic substance that must be removed from the plant. Energy from ATP is required to convert this toxic substance into a useful metabolite, such as glycerate, which can then re-enter the Calvin cycle. And this process is known as photorespiration. Now, we can measure the rate of photosynthesis using a pH indicator because carbon dioxide affects the acidity of water. We know that carbon dioxide dissolves in water to form carbonic acid. Therefore, if we were to place a water plant, for example, like Elodea, or make algal balls, which you can see in my diagram, we have got these in a solution containing a pH indicator. Now, carbon dioxide with water reacts to form carbonic acid. Carbonic acid can dissociate into hydrogen ions and hydrogen carbonate ions. So if there is more carbon dioxide, we would expect there to be a dissociation of more hydrogen ions, therefore lowering the pH, making it more acidic. If there's less CO2 in the solution, we would have fewer hydrogen ions, therefore a higher pH. We would expect there to be a more alkaline solution. So if we observe the color change over time after we have exposed these algal balls or water plants to specific intensity of light, we should see a change in the color. Now, if it has gone a purple-red color, that indicates that carbon dioxide has been uptaken by the organism and therefore photosynthesis is occurring. If the color is a yellow color, that is indicating there is a high carbon dioxide concentration in the solution, therefore photosynthesis is slow or absent. Now, this method is an indirect way of measuring carbon dioxide uptake. It's not the exact rate. For more precise measurements, we would want to use a CO2 sensor, and that would give us an actual reading of the carbon dioxide concentration. Thank you for making it to the end of my video. I hope you found it useful and helpful. And if you have, then please do like it, share it, and comment. And if you're looking for the resources that I've covered in today's lesson, then why not check out my study guide? So that is available on my website, It's available on my website, ukbiotutor.com. So go and check it out. And also, let me know how you found it.